So how's everybody today? Good? Okay. So as you have noticed, my computer is acting up a little bit. My pen is not getting recognized. I just did that update yesterday, and now it updated so much it doesn't recognize my pen. Yay. So I will try to just use, primitive ways of showing here things. I just put on the iClicker, do have some questions, so make sure you get those points, right? And also I just updated the PowerPoint, the PDF file for today. And as you noticed, I had before this nearly 90 slides on the PDF file, and yeah, it's way too much to be covered, like in one sitting, right? It's like I don't want to, overwhelm yourself or myself, right? And why did I have slides, PowerPoints like that? Well, because in medical school, we actually did cover 90 slides per sitting. Yay, and not fun. Okay, not fun for those poor guys, not fun for me either. All right, so we have now about 38 slides here, which is a lot more manageable. So we are going to continue where we left off. And we are not going to quite get to the complement system. I'm going to leave that for the next class, okay? But I was planning to start talking about it today, which I will, but just not in too many details. So I did put this little, complement picture, complete little caricature here, saying, oh, you're so strong. Oh, thanks for the compliments. Well, not that kind of compliment, right? It's like a compliment. Okay, anyway, we'll get there. So this is what you have seen before, right? Emphasizing what you need to know about this chapter, about this topic. What we're focusing here is how is it that the innate immune system is acting. So we're looking at effector functions of your innate immune system. And we know that involved in these functions, you're going to have certain cells, you're going to have certain molecules, and certain tissues like the blood vessels, they're going to be recruiting cells and acting on this inflammatory process, right? So what are the cells that are involved here? You should know that since we're talking about innate, these are the cells mainly of your myeloid pathway, myeloid lineage. So here you have those myeloid cells. And you also have this guy from your lymphoid lineage, right? This natural killer cell and some other ILCs that are working in the innate response. Okay, now what we're going to focus now is on those inflammatory responses and on the signaling pathways. So that gets to be quite interesting and sometimes a bit challenging because you do have to remember several steps and several molecules that are very important for the pathway to work. So we left off in this slide here. We're talking about those toll-like receptors, and we know there are many of them, and you should know a few of them. And you should know that their location is very much related to their function. So if you have certain toll-like receptors on the surface of the cell, they should be ready to recognize pathogens that are intact, okay? So they are going to recognize these invaders that just came across the epithelial layer or the mucosal layer, and they are, happily floating around. You have the swimming bacteria, you have some floating viruses, but they are not chopped up. They are whole. Okay, you have those fungi, right? So here, if you have these toll-like receptors on the surface here, so this is your cell membrane. I like how they always put the cell now squared, right? So here is cell membrane. So if your toll-like receptors are on the surface, well, here it's showing them as dimers, but usually they are on the surface just very close to each other, but not quite dimers, okay? Because when they dimerize, they get activated. So when you look at a cell, you don't expect to find them dimerized already. You expect to find them close to each other, but not interacting. Okay, so you expect to find them as monomers. But then here you see that those very common toll-like receptors, when they dimerize, is because they are interacting with something they recognized, right? And that I'll emphasize a little bit more in the next slide. And remember, what is it that they're recognizing? So here you see, okay, this homodimer of TLR4 is going to recognize some bacteria. So you have gram-negative bacteria, okay? And then you have this other one here, this heterodimer, TLR1 and 2, is also recognizing bacteria. And then you look at the other one, you see here, oh, TLR26 is recognizing another type of bacteria. Okay, gram-positive, but it's also bacteria. So you see a lot of redundancy, right? A lot of redundant functions for these TLRs. And you should be asking, why is it that a lot of those different TLRs are doing the same job, right? Why is that happening? Why do you think that's important? Yeah, but why? Why do you need redundancy? Quite memory, but making sure that nothing slips through, that's a good way to put it. But there's another reason. Is it regulation? Quite, because a lot of these pathways converge. Yeah, it's first line of defense. It has to recognize a lot of stuff. But if you have just, let's say, this homodimer that recognizes bacteria, it should be fine. Bacteria should be recognized. If you have many numbers of these proteins on the cells, yeah.
Let's say a bacteria is carrying another vesicle, for example, and it gets in the cell, and one receptors on the outside are recognizing?
I got confused.
Maybe the pathogen was carried on another substance, so it didn't get recognized by the external receptors.
That would be fooling the receptor. So that would be a strategy of the pathogen. But why does your cell have several receptors that recognize the same thing?
Just saying, if it fooled the outside ones, it would have the inner ones to die.
Yes, but that is in a different compartment. So I'm talking about the ones that are on the surface right now. Oh, the answer really is that if you are born with a genetic mutation in one of those receptors, then let's say TLR4 is not working. You have another receptor that does the job for TLR4, so you can survive. Okay? So if you are born with a genetic problem, that your genes just for some reason got mutated and you don't produce some of those, or just one or two of those TLRs, you are producing another one or other ones that can do the same job. So your innate immune system is going to be intact, basically. So you're fine, you can survive. Okay, so this is a very important reason for redundancy of the detection of those receptors. Okay, so here, again, most of them are homodimers, but then some of them are heterodimers, and be familiar with the ones that you should know, right? Now, putting it into logical perspective, the ones that are on the cell surface, they are going to recognize the whole 3D intact pathogen, right? Now, let's say that you this cell here is a phagocytic cell, so it engulfs the pathogen, and now you are in your phagosome or in your phagolysosome, right? So in the phagolysosome, you're going to have also the presence of some of these TLRs. They are going to recognize other things, right? Now, inside the phagolysosome, your pathogen is not intact anymore. It is getting chopped up, it's getting digested, and hopefully is getting inactivated, killed, whatever you call it, right? So here, what you want to do is you want to recognize pieces that you could not see when the pathogen was intact. outside. Okay, so inside those endosomes, you should be able to recognize like inner viral particles. Okay, so you have some viral proteins. Let's say if your virus is coated by an outer membrane, we call those enveloped viruses, right? When they get inside the cell, and I mean when the cell phagocytoses, then it breaks the outside membrane, and now there are some proteins that get exposed. So you get some TLRs that will be able to recognize those proteins. And then you have other ones that are going to recognize the genetic material of whatever was eaten. So we can have some TLRs. They're going to be looking for double-stranded RNA or single-stranded RNA with certain shapes, okay? And also you're going to have this guy here, which I kind of like him, okay? This homodimer right there, that is TLR9, and this guy is going to recognize a specific a pattern in bacterial DNA, mainly bacterial DNA. You have some viral DNA as well, depending on what kind of virus you have, right? So some of these bacterial DNA, they have some CpG islands that are not methylated. Okay, so those are CpG island, okay, cytosine guanine with a phosphate in the middle. Okay, they're not methylated. When you find these CpG islands, usually in your own DNA, they tend to be methylated. Okay, but then the bacteria or the viruses, they don't really have the mechanism to put methylation on those CpG islands. So that is a red flag for your TLR9. And how are you going to remember that TLR9 recognizes unmethylated CpG islands? And that's the fun part of it, because if you just look at the mirror image, this guy here, it's a reverse 9, right? So your CpG island here is kind of like a, you know, a dyslexic 9, kind of backwards. So that should be a little clue for you to remember that TLR9 recognizes unmethylated CpG islands on the DNA of the pathogen. Okay? All right. So here, where you find them and what they're doing, right? Remember that when they are on the surface, this is why I put this picture here from a different book. So this is from the Abbas book. Here, see, you know, monomers of those proteins. Okay, and you notice that you have this extracellular domain. Then you have this transmembrane domain, and you have intracellular domain. And that intracellular domain is going to be very important for the signal transduction that we're going to see next. Okay? So again, what you're going to find on the outside, and I know I put several slides with a bit of repetition, but then in my head it's kind of difficult to remember, you know, TLR 1, 2, 2, 4, 3, 7. So it's better if you look several times and you can get used to it. Yeah.
The endosome is relatively small compared to the host cytoplasm. So what's the cause that particle or pathogen gets through the cell membrane that makes it an endosome?
Well, the endosome is going to be involved when the pathogen is phagocytosed. Okay, so first you have to eat up the pathogen, and then the phagosome is going to get together with the lysosome, and the whole thing, you can call that an endosome or a phagolysosome. Yeah, this is mainly inside a vesicle inside of the cell. You can call that an endosome. Okay. All right, so here are some. All right, so when you look at the position of the detecting part, of the binding part of this PRR, at the surface, this thing is facing to the outside. Okay, so when this thing becomes an endosome, so of course the binding part is going to be facing to the inside. Okay, so you just follow this membrane here, like if it invaginated like this, and then it became an endosome. So you're just like bringing that thing with the detector part that is inside of the endosome. And of course, if it is floating in the cytoplasm, the thing is floating, so it's not connected to the membrane. Okay. So here, you can have your intracellular receptors, you can have some on the surface, and you can have some that are floating around. And let's see here. All right, so here a little bit more on this PAMPs and detection, which you had before as well. So just for your review here is where I put the specifics on the CpG islands, the TLR9, and TLR3 is going to detect your double-stranded viral DNA. Remember TLR7 and 8 for single-stranded RNA. Oh, why did I put that? I don't remember. Okay. So here, when you look, these guys are very interesting, the cytosolic ones, okay? So you have main ones that are nod receptors. So here put nod 1 and nod 2. You have, I hope I'm recording for a backup here. 1 second. Oh, I forgot. Okay. So you have RIG-I, or called also RIG-1 receptors, and then you have these guys here, NLRP3. Okay, so those are very, very important. So the nod receptors, nucleotide binding oligomerization domain containing protein 1. Just remember nod, all right? They are going to bind to some sugars on bacteria, okay? Now, RIG1 is going to bind RNA stuff, so viral double-stranded RNA that is floating in the cytoplasm. These are all cytosolic or cytoplasmic, floating around PRR, okay? And then you have this guy here, NLRP3, really, really cool guy. Here is saying it is binding bacterial LPS, okay, lipopolysaccharide, but that's not the only thing it binds. It can bind other stuff. Well, here is saying PAMPs, but it can also bind DAMPs. Okay, so same receptor can detect PAMPs or DAMPs, and we're going to get more into the mechanism too. Okay, so again, this is also kind of repetitive. Okay, we look at the cell surface. Okay, so soluble floating around outside of the cell. Okay, two that are very, very important ones are mannose binding lectin, and guess what it's binding? Yeah, I mean, it's right in the name, mannose binding lectin, so you can't get that wrong, right? You have ficillin. That guy's like, what in the world it is binding? Well, some type of stuff in bacteria, okay? Now, these two floating soluble PRRs are going to be very important for the activation of your complement system, okay? So keep that in the back of your mind. It's going to be very important to activate that system. All right. So here let's get to the nitty-gritties of those receptors. Now, things that detect DAMPs, damage stuff from your own cells, okay? So you have here this receptor that is called RAGE. Very appropriate name, okay? You get anybody angry, okay? It is going to bind activated glycation or advanced glycation end products. What are these guys? What is a glycation end product? When I say glycation, what do you think about? Glucose, sugar stuff, like, for example, when you, anybody cooks? Who likes to cook? Yeah, I do sometimes. Okay, I do, but when I cook, it's good. Okay, so I like to use onions, right? So I'd like to put a little bit of butter and chop some onions, and then saute the onion, and then it starts to get glycated, right? You start to see the browning of that onion. It tastes good. It tastes a lot sweeter than just the raw onion. I don't like raw onions, right? All right, it tastes better. Okay, now you can either get glycation when you are, frying some stuff, or you can get glycation at 98 degrees Fahrenheit throughout 70 or 80 years process. What am I talking about? Alcohol?
I was thinking of fermentation.
You're already thinking about drinking? No, I was thinking about your own body. 98 degrees Fahrenheit is your body temperature, and 70 or 80 years is a lifespan. Right? I mean, a lot faster than 300 degrees in 5 minutes, right? But you get the same type of glycation end products. It just takes a longer time, right? So your body naturally is going to produce some of this glycation stuff, especially in your joints. It's no wonder that older people have stiffer joints and things start hurting. Let's not talk about that, right? But it happens, right? And when those things happen and you get cell damage, then some of your innate cells might have these annoying rage receptors that can recognize those damages and cause inflammation. Yeah. Getting old sucks. Don't get old. Okay. So, inflammation. How are you going to remember rage receptor? So here is a little picture for you. There. You can get enraged about your blood sugar. Okay? So that's your mnemonic to remember your rage or your DAMP of blood sugar, glycation, right? So, then you have these other ones here. Look, notice that you're also having several receptors that can recognize the same thing or different things. In this case, you have rage that is recognizing glycation, but it can also recognize other things. Look, same receptor recognizing other things, okay? So this is not a redundant type of mechanism. This is a type of multitasking mechanism, okay? So this one is recognizing this HMGB1, lots of letters. This is a homeo box, and basically it's a protein that regulates gene expression. So that guy should be inside the nucleus, doing its job inside of the nucleus. If that guy is floating around somewhere outside of the cell, it's not supposed to be there. If it's floating around in the cytoplasm, it's not supposed to be there. So you have this PRR that recognizes that there was some damage to whatever process was happening in the nucleus and somebody escaped the nucleus, and that's a red flag. So you have RAGE and TLR2. TLR2 and TLR4 are recognizing other things too, right? They're recognizing some bacterial stuff or some virus stuff, but they can also recognize DAMP. And specifically here is recognizing this DAMP that should be in the nucleus. So if we find something out of place, that's a red flag that the cell is not working right. Okay, so those PRRs get activated and they are going to cause inflammation. And most likely they're going to cause cell death because that cell is doomed anyway. Okay, all right. So these are actually on the cell surface. Of course, if you have also things on cell surface, but you can also have PRR inside your cytoplasm to recognize those damages. Okay. Now, okay, so cell surface PRR, important cytosolic PRR. The most important one that you have is this guy here, NLRP3. Okay, yes, it has a long name. I don't care about the name. Okay, it's node-like receptor, protein 3, containing protein 3, something like that. It's type of a node receptor in the cytoplasm. Okay, it is going to recognize several things that are not supposed to be floating around in the cytoplasm, but importantly, it recognizes some crystals of uric acid that they are not supposed to be floating in the cytoplasm, okay? Because when you do have crystallization of uric acid, that means something is going on here with too much degradation of purines. Remember, purines are building blocks of DNA and RNA, okay? So if you have too much degradation of purine, you're going to have too much of this uric acid in the cytoplasm, and you're going to have a threshold that is going to reach crystallization point, and once these things crystallize, they're going to be detected by NLRP3. Okay? Now this is the main mechanism that causes gout, right, as I briefly had mentioned before, right, very, very intense and painful inflammation. Okay, and here I'm showing the joint at your big toe is always the joint at the big toe. Yeah, it can happen somewhere else, but most often it happens at the big toe, and very, very painful inflammatory process. Everybody cool with that? All right. Here comes your first question, which is, of course, now it is easy because you just looked at it. and it's also going to be easy when you take your test next week, right? Because you're going to be able to reason through these things in no time. All right, 10 seconds. Oh, we have people were shifting to the wrong answer. Just shift back to the right answer. Okay, I'm going to stop it. Okay. What is your answer? Is A. Okay, because if this thing is inside an endosome, it's going to start recognizing things of pathogens that got opened up, or broken down, right? Broken up pieces. So single-stranded RNA of the viral genome, you'll be able to see it after you break the outside capsule of the virus. All right, so here is straight from your book, just to give you that, overview on those TLRs. And no, I did not put a lot of them, so not too many. Just emphasize the ones that I mentioned. Okay, so this is from your book, and it's supposed to show you how these monomers get dimerized, but I think the book did a very poor depiction of dimerization here, because when I look at it, it did not make sense. Okay, so what is really happening here is that you have those recognition particles, okay, that can interact with your TLR, okay, but here they are not really showing how this thing is dimerized. So what you need to do is get this bacterium like right there, right here, and you can see the interaction between two domains of those bacteria, that if you have those high frequency patterns like very close to each other, then one of them interacts with one monomer and then another one with the other monomer, and they're very close together. This is when you're going to bring those two monomers to a very close proximity in order for them to be able to interact in the cytoplasm, okay, so on the other side of the membrane. Okay, if you're just floating around and something binds here and something binds on the other side over there, then you're not going to activate that. Okay, so you need to bind to this bigger thing that has a lot of those high frequency of those patterns, and then interaction of the patterns is really going to bring that thing, those monomers together. It's really going to allow the formation of the dimers. You know what I mean here? So once they are interacting, that is going to start sending the signal for the inside of the cell. You're going to start causing the cell to do something. All right. So remember, you have those two types of PRRs. You have phagocytic and you have the cell signaling ones. So here we're going to look at the cell signaling. What is it that is doing to the cell? What kind of signaling that is going to cause inflammation? Okay. So here, important signalings. You're going to have NF-kappa B, okay, NF-kappa B, and you're going to have interferon upregulation in case you bound the virus. Okay, you're going to have interferon pathway, so interferon regulatory factors, called those IRFs, and you're going to have MAP kinase pathways. Okay, so we're going to look now at NF-kappa B and see what is happening here. So here is showing you that once you have activation of this TLR4, you're going to have interferon upregulation and also cytokines. Cytokines are always upregulated, inflammatory cytokines, pro-inflammatory cytokines. All right, so here we get to this part, which if you follow from the book is going to be a bit confusing, but this here I put in the simplest way possible and hopefully it makes sense. Okay, so what happens here? PRR on the surface, it is connected, this should be inside of the cell. It is connected to something intracellular in the cytoplasm right here, and this something is a mediator molecule, or you also call it here adapter molecule. Okay, so there is something that is an adaptive molecule inside of the cell. Okay, adaptive to what? Well, to your receptor, to your PRR. So here, the most common one is this guy, MYD88. Okay, so it's connected to several of your PRRs. Okay, so what it's going to do is that once it detects activation of the PRR, because remember that activation here, you're going to have your dimer forming, right? So once you have here activation of the PRR, it's going to send a signal to that adapter protein, and that adapter protein is going to start the cascade. Okay, it's going to trigger this intracellular signaling cascade. All right. Eventually, what you want to do is that you want to get a transcription factor to go into the nucleus and start making proteins that are pro-inflammatory and antiviral. Okay? So, your cells are always ready to produce some kind of pro-inflammatory cytokine, right? So, to operate some kind of these genes. Now, of course, the cells, they have the receptor, right? So now, you can imagine that you must have this transcription factor always ready to jump into action, but it cannot be activated all the time, otherwise you're going to have inflammation all the time. So this transcription factor is held in the cytoplasm and is being held away, is not capable of going in the nucleus unless it gets a signal that it can go to the nucleus. Okay, so this is a safety mechanism that is not going to upregulate those inflammatory genes unless this thing gets the trigger and gets free from something that is inhibiting it, that is keeping it in the cytoplasm. Okay, so in the cytoplasm it's not going to do anything. So if it is in the cytoplasm and is not doing anything, it is being inhibited by something. The thing that is inhibiting it is called inhibitor of kappa B. All right, so that's easy to remember. IKB is inhibitor of kappa B. Okay, nothing happens, the thing is inhibiting, everybody is quiet. Okay, so now let's see what happens here. Once you have a PAMP, okay, PAMP that is detected, or a DAMP, in this case was a PAMP, here's what's going to happen. You're going to have your adapter protein here to get together with some other factors that are going to activate, okay, forget about these guys. They're going to activate this guy right there. That is a kinase, okay? This kinase, what it does, it is going to put some phosphate groups on your inhibitor. It actually puts 4 of those phosphate groups in your inhibitor, okay? When the kinase puts the phosphates in the inhibitor, it's going to change the conformation of that protein. Once that conformation is changed, it's going to free up. It doesn't want to interact with an F-kappa B anymore. So an F-kappa B is going to be freed up, okay? So then, let me see if I have the next figure. Here, then your NF-kappa B is going to be able to go into the nucleus. Okay, so what is happening here? You have detection of your PAMP, you have intermediate molecule that went through signaling inside of your cytoplasm and told this kinase to put phosphates on your inhibitor. Okay, what's the name of the kinase? Inhibitor of kappa B kinase. That's the IKK. Okay, so it's going to put kinase, it's going to, kinase is something that puts phosphates on molecules, right? So this guy gets there and puts phosphates on your inhibitor. The inhibitor dissociates from NF-kappa B, and now NF-kappa B can go into the nucleus and start doing its job because NF-kappa B is a transcription factor. Okay, everybody took cell bio? Yeah? Yeah, so you know what I'm talking about. Transcription factors or genetics? Who doesn't know what a transcription factor is? Okay, you can say it now or not. Okay. So, what is happening here? Here's the whole path again. This guy goes into the nucleus and you get gene transcription. That means you're going to make some proteins, right? You're going to have transcription, then your RNA is going to come to the cytoplasm, you're going to have translation, and you're going to have certain inflammatory genes and other genes that are important for this whole thing. What are the inflammatory genes that are really, really important? TNF alpha, pro-inflammatory cytokines. What is it going to do? It's going to recruit some immune cells to the site of damage. Okay, you're going to have interleukin-1 beta. This is also pro-inflammatory cytokines. It's also going to recruit some immune cells, but they do have other jobs. Okay, then you have these two guys, interleukin-6 and interleukin-8. Those are the very important inflammatory genes that get upregulated because of that NF-kappa B. Okay. Besides that, you also have cyclooxygenase 2, also known as COX-2. And this is a stress response gene that gets activated. Okay. Now, have you heard of COX-2 before? No? That produces certain molecules that cause a lot of pain and, of course, inflammation. Okay, and is a site of action of a lot of your over-the-counter non-inflammatory, non-steroidal anti-inflammatory medication like ibuprofen or aspirin. Okay, so this is a target of some of those anti-inflammatory over-the-counter meds. Okay, so COX-2, and then you have this other gene right here that is called inducible nitric oxide synthase, or INOS. It's also a stress response and is an enzyme that is going to produce nitric oxide. And that is important for vasodilation, right? Nitric oxide is important to open up some blood vessels, right? Okay, so this is the starting of inflammation. All right. So that was your NF-kappa B pathway. And now we're going to look at this other pathway also of inflammation. And this one, remember, is inside of the cell. So these are your node-like receptors. Okay. Remember, one of the most important node-like receptors is NLRP3, okay, which we're going to look at the pathway next. Okay. So what is happening here? Well, those are monomers in the cytoplasm unless they get activated. And then they become multimers, okay, so polymers of some kind of thing, and this is when you get this interesting type of structure here, okay. So cytosolic PRRs, they can detect PAMPs or DAMPs, okay. Node 1 and node 2, those are very, very important for detecting stuff that was in the bacteria, okay. And what they're going to do, they can also induce antimicrobial expression, gene expression. Besides that, they are going to start this process of autophagy. That means they are going to stimulate the cell to eat certain parts of itself and recycle some of those proteins and other and sugars and other things. Okay, so they can trigger autophagy. So let's see what is important here in this pathway. This is when you're going to start looking at these inflammasomes. Okay, so node-like receptor inflammasomes. So let's see what happens here. You're going to have here the detection of some type of DAMP or PAMP signal. You're going to have oligomerization of your monomer. Okay, in this case, let's say NLRP3, okay? And that oligomerization is going to further activate a cascade that is going to produce inflammatory cytokines, okay? And on top of everything, this thing is going to kill the cell. Okay, once you activate the NLRP3 inflammasome, that cell is doomed. It's going to be killed from the inside. Okay, you know, like that movie, I don't remember which movie that was, that has some like alien that comes in the belly and like punches through the stomach. You know which one I'm talking about? I never watched the movie, it was giving me nightmares, but I always see those memes or things like the bursting through. That's the vision you should have in mind when you look at NLRP3. It's going to make a monster inside of the cell and it's going to burst through the inside of the cell and burst the membrane of the cell. Very dramatic death. You call that kind of death programmed. Okay, so it's a type of apoptosis, but it's not apoptosis. You call it pyroptosis. Okay, it is more dramatic. All right, so here we go for the pathway. Okay, so what is happening here? You have an NLRP3 that is doing nothing, floating in the cytoplasm, just waiting to be polymerized. And here you have your lightning bolt that was able to get inside of the cell and is going to activate your NLRP3. Okay. So once this pathway is activated, you are going to upregulate certain genes, gene products. The upregulation of those gene products is really not in their active form. Okay, they are ready to be activated, but they are not at their active form yet. So these pro-inflammatory genes that are getting activated are mainly those interleukin-1 beta and interleukin-18, okay, highly inflammatory. But again, when they are first produced, they are not active yet. In order for them to become active, they need to be cleaved. They need to be cut up by this enzyme that is called caspase-1. Where are you going to find caspase-1? Well, caspase-1 is also floating in the cytoplasm, but is inactive. Is in the pro form. So procaspase-1 is not doing anything. And yeah, you have those pro genes, everybody's quiet, unless you activate NLRP3. Now we're going to see what happens here. All right. Once you activate the inflammasome, this polymer of things is going to cut procaspase-1 into caspase-1 right there. Now you're going to have your active caspase-1. Caspase-1 is going to go to those pro gene products and it's going to cut them. That's why it's called a cascade. I bump into this first one, I activate caspase-1. One molecule of caspase-1 can go around and activate a lot of other molecules of those pro-IL-1, pro-IL-18. Okay? So it's a cascade because one molecule can go here and say, oh, this guy is quiet. Let's say we have here like pro-IL-1 beta, and these guys are pro-18. And here you come with your cascade, your caspase-1 that got activated. Okay, so caspase-1 can go here and chop this guy, and chop that guy, and chop this guy, and chop the other guy, and chop and keep on chopping. Like one molecule, well, this one enzyme was able to chop a lot of the other pro molecules. This is your expanding your cascade effect. Okay, so now you get a lot of these pro-inflammatory cytokines that were activated, and they're going to be dumped outside of the cell. They're going to be exocytosed. Okay. Now here, So, all right, so here. So until now, we have really looked at the effectors of innate immune response. Okay, and what are those? Effector cells, they're gonna bind, they're gonna eat whatever they bound, right? Macrophages, phagocytic cells are effector cells, okay? And then here we also looked at some enzymes or antimicrobial peptides that can kill the cell, okay? They can punch holes in the cells. And you also have these other molecules that are called opsonins. These guys are really interesting because what they do is that they bind to microbes and they tag the microbe for the microbe to be eaten. Okay? So main effector functions of your innate cells, they have to do with directly eating the microbe or coating the microbe so it's tastier for whoever is going to eat it, okay? Or you can produce some molecules that are going to punch holes in the microbe. So these are always strategies of innate defense, and they are very effective, so we call those effectors of innate defense. Okay? All right. So now we're going to get into those objectives, and we're going to go one by one. If you're playing the Immune Quest, this is when you start getting into the complement pathways. So we're going to see the alternate pathway and the mannose binding lectin pathway, and also the classical pathway. But here, really, when you talk about innate immune system, you're talking about alternate pathway and mannose binding lectin. The classic pathway is a pathway of adaptive immune system. Okay, so it's not part of your innate immune system. You really have 3 pathways for complement pathways, okay, complement system. But things are going to get clear soon. Okay, so the tricky thing about this immune response is that everything is so interconnected that it's like a kind of tricky to talk about one thing without talking about another thing. So I'm trying to put it in a flow that would be making sense. Okay. So we have started looking at the inflammatory process. We look at some pro-inflammatory cytokines, and now we're going to see how is it that those molecules are triggering this whole inflammatory process in general. Okay. So we know that this acute type of inflammation has to be very fast, right? And what is it going to do? First is going to recruit cells. They're going to help you kill the pathogen that invaded the tissue. Then it's going to try to minimize the damage and try to isolate the pathogen to one specific area, okay? And let's see how this is happening here. So remember those pro-inflammatory cytokines that happen because of that TLR or NLRP3, some kind of PRR signaling, produce those pro-inflammatory cytokines. And those got dumped outside of the cell. That means they got into the circulation. They got to your blood. In the blood, they're going to go all around, all over your body, right? They're going to reach your brain. They're going to reach a part of your brain that is your hypothalamus. And if there is enough concentration of those pro-inflammatory cytokines, they're going to cause fever, okay? Now, they're going to increase your body temperature in general, okay? Besides that, they're going to go all the way to your liver. And in the liver, we are going to trigger upregulation of certain inflammatory proteins. These are C-reactive protein, okay, CRP. Also, look at this guy, complement protein, complement 3. So that's your C3 protein. This is the most important protein in the complement pathways, all of them. All 3 pathways. This is the converging, most important protein of complement system, okay? And you're also going to upregulate this guy here, fibrinogen. Fibrinogen is something really important for blood clotting. When you see a protein that ends up in ogen, it means that it's not active, so it is quiet. You want it to be quiet. right? Unless you have some damage to some blood vessels, because if that happens, you could have internal bleeding. You could die, right? So in that case, you want to have enough stock of your precursor protein in case you have to go to the damaged blood vessel and cause coagulation, right? So fibrinogen is very important for part of that coagulation cascade, okay, to plug holes that happen in your blood vessels, okay? So these three proteins are upregulated once those pro-inflammatory cytokines reach your liver, okay? All right. So here you have your cytokines. We talk about effectors, right? We have now your cytokines, and you're going to have those other proteins that are also going to help with your inflammation process. Besides all that, remember that certain cells of your immune system are able of producing interferons. Besides that, all of your nucleated cells, all the cells in your body that have a nucleus, okay, are going to produce type 1 interferon, okay, all of them, okay? And you're also going to have those other proteins that are going to help with inflammation and with opsonization of pathogens, with tagging the pathogen. Okay, so what are the signs of inflammation? Well, I'm sure that everybody has gone through some kind of inflammation, right? Here it's showing you the finger because, you know, some of us kind of like whenever we get anxious, we start biting nails or biting cuticles, and sometimes you get a little, you know, inflammation or bleeding. You know, I'm talking about the side of the nail, and then it hurts like hell, and you're like, oh man. So what are the signs of your of inflammation? First thing, it gets hot, right? The thing gets, yeah, the place gets hot. It's gonna become red. Why do you think it gets red? Blood flow, yeah. You get some vasodilators, right? Vasodilators that are going to increase the blood flow to the area. So it gets red. And it swells. Yeah, you get edema. Okay, edema is the other name for swelling, okay? Because you have some fluid that's going to leak out of your blood vessels and other vessels, okay? It's painful. Yeah, because inflammation can happen not only because of infections, but can also happen because of tissue damage, right? If you like to work with, I don't know, some kind of, you know, home project, and you're like making a little birdhouse, and you're hammering these things together, and you miss your aim, and you hammer your finger, what happens? It's a simple question. It hurts. It hurts. Don't try it at home. Believe me, it hurts, right? It hurts. You damage the tissue, you're going to have damage-associated molecular patterns. They're going to cause inflammation. You're going to have redness. You're going to have a swollen finger, right? You're going to have a pain. And you have this, you know, these are the classical signs of inflammation. And then you have this fifth sign of inflammation that is temporary loss of function. What does that mean? That means if you have damaged finger, most likely you will not want to use it because it's going to be too painful for you to use it. So it's like temporary loss of function of whatever is inflamed. Okay. All right. So when you talk about acute inflammation, you should think about neutrophils, because those are the main mediators of acute inflammation. Okay. Now, what is the hallmark sign of acute inflammation? Okay, what happens is that it is happening really, really fast, but then it resolves relatively fast as well. So you get inflammation, and then you get tissue repair, and thing goes away like nothing happened. So that is acute inflammation. In comparison to chronic inflammation, which is not normal, chronic inflammation is really a pathology, okay? What happens here is that you have different cells now that come into play for chronic inflammation. So those are mainly going to be T cells and some macrophage type, okay, but mainly T cells. What happens here is that in comparison to acute inflammation, okay, so let's see what happened here. You have the cause of the inflammation, and then let's say if it was a bacterium, right? So you recruited all those cells, you went through this whole process, and you killed the bacterium disappeared, inflammation is gone. In case of chronic inflammation, you have this onset that it doesn't go away, like ever. So if it is a bacterium, you can't get rid of it. If it is like uric acid crystals, and you also couldn't get rid of it, there are treatments for that, by the way, but here it keeps on going forever. So the chronic inflammation only gets worse as time progresses. It will never resolve, okay? If you don't take care of it. Sometimes you can't take care of it, but natural process of this is just getting worse and worse and worse, okay? So this is pathology. This is normal course of things, okay? And the cells that are most associated with pathological chronic inflammation, those are really T cells. All right. Again, so acute inflammation is something that happens normally, and then chronic inflammation is really pathological. Okay. So cells that are involved in your acute inflammation, mainly your myeloid cell lineage, okay? But you also get your natural killer cells that can be involved in there, but mainly your innate immune cells. Molecules that are very important for your acute inflammation, okay? Histamine molecules. Histamine can give you allergic reactions, right? And it can also dilate your blood vessels. So histamine is an important one to know, right? Am I running out of time? No, I still have some time. Okay. Then you have prostaglandins and leukotrienes, right? Those are painful molecules. They're going to cause a lot of pain, okay? And of course, you have your pro-inflammatory cytokines, right? Okay, here what can cause inflammation? We already mentioned that, right? You have infection, you have damage to the tissue, you get some toxins that can cause very severe inflammation, right? Or some substances or some particles that penetrate your skin also can cause inflammation. And consequences, right, of acute inflammation, remember your pro-inflammatory cytokines, and here you have those proteins that were produced by the liver. Okay, so now you're going to know what they are in more details. You have those proteins of your complement system. C3, most important one. You also have C4 and C5. Those guys, they are also called anaphylotoxins. They can attract immune cells to the site of inflammation. Okay? Right, mediators, we looked up some of these chemicals, right? I mentioned those are very important for vasodilation and pain, okay? Your neutrophils, when you think acute inflammation, you should think neutrophil, right? Now we're going to look more into this function of the neutrophil, right? And remember, I mentioned vasodilation, right? And histamine is one of the factors that can cause vasodilation. And of course, edema. Edema is also synonym for increased vessel permeability. Okay, we're going to look in more details at that too. All right. So here, another picture of a foreign object going through the skin and causing some inflammation. Okay, so you can just look through this and review, right? So let's see here. Those are the effectors, right, of inflammation and innate response. So let's look at what is happening to this blood vessel at the site of inflammation. Okay. So first thing that is going to happen here, you are going to have at this site where you have the damage or the infection, you're going to have production of those pro-inflammatory cytokines and also of histamine. Let's focus here on histamine. Okay. Histamine is going to come in contact with your blood vessel. This is your blood vessel right there, okay? And what it's going to do is going to increase the permeability of that blood vessel and is going to dilate the diameter of that blood vessel. Okay, so two things are happening here to the blood vessel. First thing is that the little separation between the cells, in endothelial cells here of your blood vessels, that separation gets bigger. If you get bigger separation here, that means stuff that is inside of the blood vessel can leak out. This is why you get edema, swelling, okay? Histamine, prostaglandins, those kinds of pro-inflammatory molecules are going to open up spaces in the blood vessels and allow liquid to flow out of it, okay? Besides that, the blood vessels get bigger, okay, so that's dilation of the blood vessel. Remember that you also have here your eye nose that makes nitric oxide, another thing that's going to dilate your blood vessel. Okay, so everything is happening here at one time. Besides that, so remember that stuff happening with blood vessel. Besides that, the cells of the blood vessel, they're going to start upregulating certain types of molecules on their surface. Those molecules are sticky molecules. And they are sticky molecules of endothelial tissue. That's why you call them like E for endothelium, E-selectin. That's a good one to remember. Okay. Now, why is this important? Well, because now you have to think of what's happening here in the blood vessel. What's happening in the blood vessel? Okay, let's pump some blood into the brain. What is happening with your blood vessels? Inside the blood vessel. All right, chocolate. Still have time. Hopefully I have, yes, I have one chocolate left. Lucky person from the back. Inside the blood vessel? No. I already said that. No. Okay, I want people in the back. They're really, really quiet. Are you guys awake? Just have a few minutes. I want to see what's happening inside the blood vessel. Come on, guys. This is like, okay. Everybody awake? Yeah? Hopefully? I was just listening before I started my lecture, like you guys were talking about some other professor, hopefully not me. That guy is boring. Oh my God, I wonder what they talk about me after the class. I don't want to know, okay? I feel my ears. All right. Somebody in the back. What is in your blood vessels? Oh, with the chocolate. I want something simpler than that. Huh? Who said that? Yes, blood. What is inside of the blood vessels? Blood. And how is that blood flowing? It is flowing. Hopefully, if you're alive and your heart is pumping, your blood is flowing, right? If your blood is flowing at a certain speed, when you get, let's think about the blood. What's in the blood? Review for test. What's in the blood? Cells, right? So you got cells, red blood cells, white blood cells that are flowing. How are those cells going to know that they have to go to the site of inflammation to kill off some invader? They are not going to know because they are in a highway of blood and they're going to keep on flowing. If nothing tells them, hey, stop here and come out, they don't care. They keep on flowing, right? So imagine this is the highway of blood cells. And this is I-95, okay? So highway of blood cells. This I-95 has exits from both sides. So if the exit is on this side here, if you want to get to the exit, you cannot be going at the same speed as you are in the fast lane. It's just not practical, right? Let's picture here what is happening. Like, let's say, for example, you are driving on I-95, and you know you have to go and pick up a friend that is on exit 42, right? And you are driving right in the middle of this highway, and then you see your friend over there, and you say, oh, hi, bye, sorry. You're not going to be effective of going to pick up your friend, right? So you need to have certain strategies here. The first thing you should think about is, oh, I'm getting closer to my exit. What is it that you do normally if you're a good driver on I-95? You slow down, you go to the right, right? No. Have you seen these crazy people? They just cut in front of you, they're missing the exit. Oh my God. It's like, And you slow down, and then you exit peacefully, and you go meet your friend, right? Same thing here, okay? So picture this. This is important concept. What is going to make those blood cells, right, those white blood cells to slow down? It's really adhesion molecules on the blood vessels. Because now with inflammation, you start upregulating those adhesion molecules. They are going to interact with other adhesion molecules that are in your white blood cells. So now this is like Velcro. They start sticking to the side. If they stick to the side, they will slow down. Okay? Important ones to know: E-selectin, P-selectin, ICAM1. Can I slow you down? I can. Okay, ICAM1. And what is the thing is going to interact with? LFA1. Okay, this guy here is going to interact, is going to slow down. Okay, once you slow down and you get this interaction, you're going to trigger your white blood cell to squeeze through that little opening. So you better have a little opening over there, because if you do not have those inflammatory cytokines and molecules here, then this opening is going to be too tight, and your white blood cell is not going to go through. You don't want white blood cells to go through. That's the hemorrhage. White blood cells should go through. Okay, this is a process of diapedesis, and you should know diapedesis. So you should know what's happening here. First thing that is happening is capture of your floating white blood cell. Then this thing starts to roll on the side here with adhesion molecules that are interacting and slowing down. So you call that process rolling, of course. Once you do your rolling and your adhesion molecules are interacting, then you go through diapedesis. And even that way, you still don't know where to go. You have to follow a trail of increasing concentration of cytokines to get to the site of inflammation. So if your bacterium is like right here and you did diapedesis right there, you are going to follow a trail of increasing concentration of cytokines, and you're going to follow that to get to your inflammation site and to eat the bacteria. That process is called chemotaxic migration. Yeah.
So when those monocytes are pulled out of the blood vessels here and they go into the tissues, is that when they differentiate into macrophages?
No, that's a different process. Those are neutrophils here. The differentiation to macrophage is just a process that happens all the time for continuous surveillance. But these, you're activating the guys to go there and do the job. These are you're activating your effector cells. Okay? All right, so everybody cool with this? Okay. And now, yeah, now I'm out of time. So here, rolling, diapedesis, chemotaxic migration. And this is a picture you should be familiar with because it's going to be in your test. Am I telling you what I'm going to ask you? Yeah. Okay, so I'm going to stop here. Just to conclude for these last two slides, we're going to start the class on Tuesday with the extra credit points with this question. And here is just the last slide on inflammation. And these are things that I have already mentioned in the class on the signs of inflammation. I also mentioned that you have over-the-counter non-steroidal anti-inflammatory drugs that are important for inhibiting the inflammation and inhibiting these enzymes. There are COX, especially a COX-2. The name of the enzyme is cyclooxygenase. Okay, so this enzyme causes production of some byproducts that cause pain and further inflammation. Okay, what is it that you need to know here? the resolution of inflammation. Of course, things come back to normal. Remember, neutrophils are the very important leukocytes for acute inflammation, and they don't last for a long time, so they are always with a high turnover. Okay. Remember, those get recruited at the blood vessel, right, by upregulation of adhesion molecules. And let me see. All right, so at the end of the inflammation, you're going to have some scarring or some fibrosis that can happen. But then everything goes back to normal situation if you just have good resolution. And remember that I mentioned for chronic inflammation, this is a pathological condition, and it never goes away, basically because what is causing the inflammation, the chronic inflammation, the agent that it causes it just never goes away. So you keep on having stimulus of the inflammation. And again, the important cells for chronic inflammation, those are really your T cells. That was it for this lecture.